Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
First of all, I'd like to thank the organizers to invite us and to give what I think is going to be a very unusual talk, unlike everything else you heard before. I'd like to start with introducing our team, and we thought that the WORM team is a very appropriate name because even though we do very different things, it has meanings both for soil ecologists and for computer scientists, but ours, of course, is much more benign. So here are the computer people. Andreas is here with me back there, and Razvan and Sam are his graduate students at the lead guitar, and database expert and astronomer, astrophysicist really, Alex Sully. We have an undergraduate. He's the only paid employee in this case for this project. Josh, who is a physics major, he's a sophomore and helps a lot with the calibration. And I'm a soil ecologist, so if you would like to know why the earthworms are coming out or not coming out right now, as it's raining outside, I can answer that. I can also answer all sorts of questions about how long they live, how they mate, and things like that. For software and hardware questions, there's Andreas back there. So it is a sin in soil science to pull soil dirt. Many people do that. We do not do that. The reason being is that soil is an entity of biological origin, and soil is a living thing. Soil does tremendous services for us. Its significance is enormous. It provides support for plant life. It's habitat for organisms. And there are a lot of issues with carbon storage, elemental release, and water storage. This is something people usually don't think about that makes soil science very exciting and significant. Despite of all this, we know very little about actually how the soil system works, and this is why this issue of science about a year ago was devoted to these questions and why they call soils the final frontier. It is also called the poor man's rainforest, and the reason for that is soil harbors enormous variety of life, and most people don't realize that. But if you want to go out here in the forest and sort of lift up a handful of soil, you find more creatures in there than in a handful of soil in the actual rainforest. It has a lot of biomass. Again, we don't think about that, but you can imagine that a tree, for instance, has as much biomass above ground as below ground, or I should say the other way around. Some plants, grasses, have actually more biomass below ground than above ground. So there's a lot of activity going on under our feet. And this quote comes from E.O. Wilson. You might know who he is. He introduced the term biodiversity. And what it refers to is that the organisms that live around us, the tiny little ones, provide us certain services for us humans, but also in general for the ecosystems, without which life would be very different. And this is why he called, in his book, In Search of Nature, soil organisms, or invertebrates in general, the little things that run the world. And this is just an example of that. This actually is a soil sample that comes from Canada, and it was one of the group of these little things. These are soil mites. And they were extracted from the top 10 centimeters of a meter by a meter area. And you see 200 different species on this picture, actually more than 200. So why are these organisms important? Well, one of the most important roles that they play is that they mediate the rate and the pathways of organic matter decomposition and nutrient release. Because microorganisms cannot move around, these soil organisms do that for them by tunneling and generally just moving on the soil surface or moving up and down in the third dimension. They change the soil characteristics, obviously hydrology, the pH, because again they create structure in the soil or on the soil surface. They're food for organisms, and they also, some of them like centipedes and spiders, another incredibly diverse group, they can control prey populations, which can be a good thing or a bad thing. So the reason why we don't know much about how the soil ecosystem works, one of the reasons is this enormous diversity of life, and because we work with organisms we don't normally see. However, just as important is those 2 points down in red, the enormous spatial and temporal heterogeneity of the soil system. And we heard about heterogeneity earlier. Soil is the most spatially complex part of any terrestrial ecosystem. And if you look at, for instance, you take a cube of soil here, you can see all these various structures, microhabitats, occupied by different organisms. If you just take out a tiny little one of them, it's just as complex and just as diverse. So we're dealing with heterogeneity, or we have to deal with heterogeneity and complexity somehow, and it's very challenging. And I'm just giving you 3 examples of organisms which create these heterogeneities and different spatial scales. This is 2 centimeters, so these creatures work on, say, 10, 15 centimeters. Earthworms build longer tunnels or Y-shaped burrows in sometimes 1 to 2 meter deep. And then this is an excavated ant nest, and you can see how complex it is. And this kind of heterogeneity, of course, is a problem, but the heterogeneity can be the question, and in many cases for us this is the question. The soil biologists might want to know what are the microclimatic conditions in each of those chambers, and how does that, for instance, relate to hatching success of the eggs or survival of the larvae or something like that. Or you might want to know what is the microbial activity in these earthworm burrows, because we do know that earthworms generally enhance bacterial activity. And I want to have a handle of that, and I want to be able to measure that. Another example of the heterogeneity to the 2 other dimensions, I work in urban ecosystems. And by the way, urban ecosystems are the most heterogeneous ecosystems compared to any other sort of natural ecosystems, because we have all these man-made patches of grasses and trees and impervious surfaces and forested patches and things like that. So this just shows the distribution of earthworms in terms of their density in a grid like that. This is about, I don't know, if these points are about 10 meters apart. And these circles indicate high earthworm density. And we do have absolutely no idea why they form these patches. And what's important, or even more important, why these different patches are occupied, invaded, in fact, by different species. So we want to know something about that environment. Another example, I'm interested in invasive species ecology, and that's a hot topic. It's a global environmental issue, or another global environmental issue. Most of you don't know, but in North America, with the exception of the southern and some of the western areas, the earthworms are coming from Europe and Asia. We do not have native species in certain areas, and in fact these non-native earthworms are invading into forests and changing how these forests function. So we want to understand why this is the case, and one of the reasons for that is in the biology of the species. So certain species are just more successful because the way they reproduce or whatever their physiological tolerances are. But the other part of the story is in the environment and how we humans modify the environment. And here's an example of that. These are a cross-section of a stream bed in what would be a very typical natural stream channel, which you normally find in rural areas. And riparian just means the habitat along the streams. So what you see here is that the water table is very close to the surface. You have a very natural floodplain, so that's where the water spreads out after a rainstorm or something like that. You have wetland plants. You have very wet, highly organic, moist soil, sort of mucky kind of soil. And that's the habitat of this native species, which is one of the largest, at least in the North Atlantic. Now, in an urban stream, what happens is because you have a lot of impervious surfaces, a lot of runoff, and also because of the engineering activities of the stream beds, it looks very differently. You basically, the floodplain disappears. It's very highly incised. The water table is lower down. This is a much drier environment. And this is an environment in which that species can no longer exist. So it gets replaced by this Asian snake worm. Now, it would be great for us to be able to monitor where the water table is in many different places over long periods of time to have an idea what happens with the water table after a storm event and so on. So this is where the perhaps the sensor networks can be useful for us. It could provide us continuous data set in what, and that's the important part here, biologically meaningful scale. So I would like to tell how many sensors I need, how I want to space them, how frequently I want to sample, and things like that. And in the soil biologically meaningful, as you can see, just basically ranges over a lot of orders of magnitude. And it is, again, because of the organism. So some of the activity happens on the bacterial scale and others on larger scale. So depending your question will determine what the scale of the study should be. Obviously, it means less disturbance. And for soil ecologists, it's important because even if you have a technician going to the field and, say, taking the data once a week, what you do, you walk on the soil, you basically compact the soil, you can see where people are walking, you disturb the organisms, the vegetation, you don't want to do that. Less accessible sites. For instance, if you work on wetlands and wetland soil, you can't even walk on those. And even, this is our group here, getting to our field site, which involves jumping over a fence. Some of us did it more gracefully than others. And so you don't want to do that even once a week. And of course, theoretically, it requires less labor. I'll get back to that. So we built a very simple system this summer, basically, what you see here. And these are the specifics of the hardware. We used MyCases because we wanted to connect them to other sensors. In this case, it's very simple. We have soil temperature and soil moisture sensors, which you can see there. And we built everything from scratch, including the boxes, which we tested. It's called a bathtub test. We submerged them and then froze them and then melted them, things like that, just to make sure they are waterproof. We deploy these next to the building where I work. And what you see here is basically, this is a forested site. That's just a settling pond, a wetland. A stream goes through it. And there's a nice slope. So you create basically a nice moisture gradient. And this is my office here. This just describes the network design. We had 10 modes, and this is how we place them, about 2 meters apart. Maybe I should come in here. We stored the samples in flash because we didn't want to lose the data. And then we transmitted the data. That's the other reason why we tested this next to my building to a dedicated computer in my office, or we wanted to do so anyway. So the raw data, we basically collected the raw data, and these raw data were entered to a database, and you can read the specifics of the database here. We adopted or modified or utilized the technique or the methodology that was developed for Alex Salai and Jim Gray for a much larger data set, namely the Digital Sky Survey, the SDSS project. And basically, the idea of the database, of course, that in addition to storing the raw data persistently, and we do want the raw data. We actually don't want anything to convert or anything like that. We want the raw data for a number of reasons. We do not want to lose anything. So the purpose of the database, or the role of the database, in addition to storing this data, is to perform various conversions and calibration. And I'm going to show that to you right now. So again, we get the raw data from the modes. And these are the raw data that are arriving from the modes. And none of these, with the exception of the light intensity data, everything that's going to be useful or final for us is shown in purple or pink. None of these is useful or says anything to me yet. Now, these data are then converted into voltages, and there has to be a lot of modification and conversion using all the calibrations that we did before we deployed the sensors. So for instance, because we had to build in a resistor to the modes themselves, we had to calibrate for that. We also, because the voltage is changing as the battery is getting worn out, of course we have to take that into account. And then we calibrated the sensors themselves so that I can actually believe that what those individual sensors show eventually will be good data for me. Every mode and every sensor was individually calibrated. And then we had to calibrate the moisture sensor, but in addition to calibrate that in the lab, we also had to take into account that they shift with the soil moisture, so we have to take that into account and convert the data accordingly. And then I get a value, water deficit and soil matrix potential, which is very useful for people who build hydrology models or plant ecologists. But for me, I need the data in volumetric water content. So we have to do another conversion and another calibration, or we have to have some sort of relationship there. And then obviously we have to express the air temperature in Celsius and the time in some useful time form. And all of this is being done in the database. So we actually got some data, which was really good. This data set shows air temperatures in blue and soil temperatures in brown, and the light-dark cycle during a 2-week period. And it does at least validate that the sensors are working. Obviously, soil temperature is not changing as much as air temperature, but we can also see a slightly decreasing trend as we move into the fall. The event up there, I just want to mention, we had to take up the modes at some point for reprogramming. And if you look at just data for one 24-hour period, just for one day, we can also see some interesting things. For instance, this sensor was showing almost 1 degree Celsius higher value during this period of time. And if I hadn't done if we hadn't done all these calibration and conversion and so on, I wouldn't know if this is just a strange behavior of this sensor or this is real data. And I really want to know, because I want to detect heterogeneity in this case. I want to know if what I put in the different places showing different values because those places are different. Okay? It also shows that the highest temperature we reached not at 2 o'clock than normally people think, but a little bit later, which is due to the fact that this building or where the place where they are deployed is of northern exposure. So in a way, it sort of validates that what we're doing is sort of showing something. Now, these are data for the moisture sensors, again for the same 24-hour period. These are, at this point, the moisture data are expressed in pressure units. The higher this value, or the closer they are to 0, the more moist conditions they are indicating. These are just data for 4 moistures, for sensors. Now, what it shows is that there is indeed a nice gradient. The dark blue and the purple, which are closer to the stream, showing higher value. Basically, the blue was sitting in a saturated soil the whole time. It also shows that because we had to put the sensors in a saturated conditions, It shows that they are drying out, and it takes about a week for them to equilibrate. This orange one is just doing just that, very slowly. And the light blue one was sitting on the driest spot, and so it dried out much more quickly. Now, this happened to be, because we want to capture extreme events, of course, that's always an interest of ecologists. That's why we want continuous data set. It happened to be a record dry period in September when we did that, so we sort of artificially created an extreme event by just pouring 40 ounces of water around this moisture sensor, and it reacted to that. But if you don't have a continuously collecting data set, we would not be able to capture that. So we collected about 200,000 data points during this 2-week period. I think testing and calibration for this kind of work is absolutely essential. And in terms of the labor, saving labors, we're not there. I mean, basically, there was a constant involvement of mostly the hardware and software people to fidget with these. And it is because we are one of those, where is he walked out, the keynote speaker, academically dull applications, and nobody is doing that thing for us. So in terms of the wireless network sensor network side, we had good results and bad news, where we learned things that we learned from. The biggest problem was the transmission of the data, and we have data on that. Andreas is going to show some slides tomorrow. So next step, we would like, obviously, to expand on that, add more sensors, different instruments. And because, again, I'm working in an urban environment, we want to deploy them in multiple sites. We want to compare urban forest, for instance, to rural forest. And I just want to mention that in terms of environmental sensing, there are many different ways of doing that. There will be sites with a lot of money, a lot of expertise, staff, resources to do very state-of-the-art cool stuff. But at the same time, there's a potential pool of users out there, field stations or colleges or high school, who would want to know, say, how healthy their stream is in their backyards. They do not have either the resources or the expertise or the interest to do very complicated things. So they need cheap, reliable, and still accurate measurements. So we were able to capture this heterogeneity, what we set out to do. We got some data, and potentially that's going to change the soil ecology. We were very inspired by this technology and still excited by that. But again, we do feel that we are perspiring very profusely. I mean, it is a very hard work at this point. I also want to mention there were $1 million numbers flying around in terms of the budget. Our budget is $25,000, $20,000 of which is coming from Microsoft Research, and we spent half of it. None of us is working on this full time. In fact, for the most of us, this is a recreational activity. So we're very grateful for Microsoft for supporting us. Wei gave us the first boards to play with, and Tom, who was very helpful, giving us technical information about the watermark sensors. Thank you.
So thank you. This is very interesting. It's nice to hear from sort of what I would call a real scientist, someone who's trying to use this technology. But it's clear that you ran into a lot of issues that I think a lot of us are kind of keep stumbling into. And part of what Chris Peaster was saying this morning has to do with he really wanted to just give something to someone that would just work.
Yeah.
You talked a little bit about the calibration and the testing of the sensors. And I think this is something that we need to do a lot more work on ourselves, because from your own perspective, when you look at the data, you need to know whether it's real or there's artifacts. So you have a ground truth in the ground, not in the lab, but I mean in the ground with the system for your
go through and say, are the wireless sensors collecting the same data we would? Or let me ask this another way. Do you have another way of collecting the data that's not wireless that you can compare this to?
Yeah, so what we did for now, just for the soil moistures, we used, there are many different ways of measuring soil moisture, and none of those give the same results, unfortunately, because they all are operating in different principles. But we did at least went out. There is one which called a theta probe, which you basically stick into the ground and walk around and then sort of get the measurements in a handheld device. You can do the same thing with the watermark. It does. You get a handheld device, and we did that. And so we tried to correlate the two. But because, for instance, the theta probe works on a different principle, it will give you slightly different, or sometimes not so slightly different, final results. And so you can correlate them, but at least it shows the same trend, but it's not going to give you the same exact numbers. Yeah.
So you mentioned that in practice there was a lot of fiddling with the nodes and
Oh, yeah.
how they work. If you were to classify it, were it sensor firms, radio communication firms, or software firms? That's what's the main difference.
So I would say software problems, bugs, the problems, and radio problems. So we thought because of the range that we would be able to gather most of the measurements on a single hop. This turned out not to be the case, and had to do basically with the slope, a big slope, and also lots of multiple levels of foliage. So we were not able to gather most of the data. In some cases, we had to actually go there with a laptop to communicate. But actually, in terms of sensors, they were very reliable, and the enclosure worked perfectly. Actually, some that we don't have here, the 2 days, last week actually was a flood warning in Baltimore, so lots of rain, but actually we inspected them and all of them were very well.
We don't know about the data until we go back, but they weren't washed away, I can say. So that's good.
Joe?
So one of the reasons, as Cam pointed out, is that we wanted to gather all the data. So what we do is that's why all the data is collected in Flash. And so then we have a very confident amount of data will be lost, because of the problems of the data channel. So that's why we went, coupled with the fact that we had our own custom sensors, which we had to write into the low-level code, we went ahead and
If I had an option to have wires.
If you have an option to have wires,
So, okay, so this might come up next time. Wires are very dangerous in nature because mice are coming and are going to chew on them. These are things that, now seriously, if you have wires, you have to protect them. The other problem is, for instance, if you don't have wires, it's less conspicuous. And if you work, for instance, in urban ecosystem, people won't notice that. They want to know what it is. They pick it up or dig it out and they destroy it. So there's a lot of problems associated with that. Also, again, I also work in a wetland. When you go this deep down into the mud, I mean, you can't walk in there. And to have wires 200-300 meters from the shore is just not an option, really. So.
Okay.